Hello everyone, my name is Dogbey and I'll be presenting on modulation of engineered viral vectors for targeted delivery of monoclonal antibodies. I'm a PhD student from University of Cape Town. Okay, so as a way of introduction, gene delivery systems are constructs which delivers therapeutic biomaterials to disease cells. In that, therapeutic strategies such as cancer gene therapy and immunotherapy have largely depends on this form of delivery vectors for them to carry out therapeutic and diagnostics to target cells. But for solid tumors, the aim is to penetrate the tumor microenvironment, which has been a major hurdles for this form of therapies in order to effect cytotoxicity. So this form of therapies make use of treatments such as modes such as ex vivo and in vivo models, where in vivo where cells are taken injection of viral or non-viral vectors containing the therapeutic, and ex vivo where cells are taken from the patient, modified and injected back to the patient. Now for solid tumors, the tumor microenvironment has been an impediment to such treatment strategies. For example, the use of naked monoclonal antibodies, ADCs, and bispecific has largely failed to yield good results in solid tumors, such that they are unable to penetrate the core of the tumor to effect cytotoxicity. So we thought wise that viral vectors, for example, the adeno-associated virus, which is known to easily transduce wide range of cells, post-mitotic cells, including cells of the central nervous system, we thought so of coupling the surface of such viral vectors with these monoclonal antibodies so as to able to penetrate through the tumor microenvironment and elicit their cytotoxic potentials. So emerging evidence is showing that the ideal targeted viral gene delivery system should be that have genetic modifications where the capsid is modified for tumor targeting, for improved transduction. There should be a chemical conjugating where bioconjugating strategies will be used to couple the surface of the virus or the genome of the virus with cytotoxic payload or fluorophores using protein chemistry or rational protein engineering. Then the third component should be this surface exposed monoclonal antibody or suicide gene or cytotoxic payload should be able to effect the activities as to why they've been delivered to these tumors. As such, evidence emerging from Mervel and et al. In 2020, for example, make use of all these three mechanisms such that it has resulted into a good outcome. Okay, in our lab here in UCT, we are specialized mainly in Snapdrag technology, where Snapdrag is a genetically modified human alkyl guanine DNA alkyl transferase, a monomeric variant that forms a covalent coupling with a benzyl guanine protein derivatives, such that any diagnostic or cytotoxic protein that's labeled with BG can easily conjugate to the Snapdrag in order to be delivered into the cognate cell surface receptors. And proof of concept studies over the years have shown successful use of this technology in targeting EpCAM, EGFR, CSPG4, CD64, among others. So a little look at the Snapdrag technology, as I said earlier, is a human-based protein which form a covalent coupling with benzyl guanine. So once it's reacted, the guanine version is dropped for the labeling with any benzyl guanine modified substrate. It could be a cytotoxic payload or a diagnostic fluorophore or a reporter protein, such that it results into a 1:1 stoichiometry. So a combination of these two leads to a very higher product. Okay, so that's as a way of introduction. As such, I thought as much to start my experimental procedure to date. To date, I was able to knock out the native binding motifs on the adeno-associated virus such that once it's been delivered to in vivo models, immunogenicity, which has been the major hurdles of such viruses, will be eliminated. They went ahead to do the triple transfection in hex cells, where plasmid bearing the transgene, the capsid, and adeno helper virus plasmid were transfected in hex cells and followed up for about 5 days, where cell culture supernatant were collected every 24 hours. And this image just shows 72 hours post transfection of successfully expressing of these hex cells, showing the EGFP reporter protein encoded on the plasmid. This shows I went ahead to purify this virus for a downstream experiments. So on the other hand, I'm also involved in expression, production, and purification of the single chain variable fragment, where anti-EGFR scFvs plasmid were transfected in mammalian cells. Cell culture supernatant were collected every 3 to 4 days for about a month. Then I went ahead to purify this on the IMAC protocol, such that these recombinant proteins have histag-based sequence in them, as they are able to bind to the nickel column on the IMAC protein we have in-house, and I was able to yield a very high amount of protein, which would be conjugated to the surface of the virus for delivery into receptor positive cell lines and in vivo models. So my current ongoing work is to make sure I conjugate the monoclonal antibody to the surface of the virus. This I will be using the click chemistry conjugation method, such that the viral vectors have specialized functional motifs on the surface, spread across the surface of the virus. As such, the viral particles, as such, the conjugating of this monoclonal antibody will be able to deliver a product which have one virus to several monoclonal antibody exposed on the surface. Then we'll go ahead to do quality control to determine how much protein and conjugating to the virus, do functional analysis, and determine the presence of the virus on SDS page. Then once that is confirmed, I will now go ahead to do in vivo evaluation, where EGFR positive cell lines, specifically A431 cell lines, will be used in this evaluation. Then now proceed to in vivo models. So as a way of conclusion, preliminary data that we've generated in the lab have provided a supportive evidence for a targeted AAV drug delivery platform, where viruses which easily transduce tumor models or transduce wide range of cells will be used as a delivery vector. Secondly, will also serve as a novel platform integrating cancer gene therapy and immunotherapy. Now, the advantage of this model actually is that the non-viral delivery platforms failed to transduce tumor cells. As such, when we use AAV, which are easily transduced cells, different wide range of cells carrying this payload, it's easy to elicit cytotoxic effects in these tumor cells. So as an ongoing work, I would like to thank my professors and my funders for the general support of this work as we go ahead to produce a very good result in the coming years to come. Thanks everyone, and I'll be on standby for any question. Thank you very much.